myself now. You guys are not being recorded. I'm not doing recording the Google Meet. I'm just streaming straight to YouTube and I'll post it later. Okay, warm up. All right, so I showed you a lot of these warm ups have review things. So some of this is unit 0. But 41, which macromolecule consists of glycerol and 3 fatty acids? That's an easy one. All right, somebody type it in the box for me. Glycerol and 3 fatty acids.
A lipid.
Yes, lipid. Thank you, Leo. 42, a substance that prevents sudden changes in pH. Anybody remember what that thing is called that will get an acid or a base back to neutral? Starts with a B. Yes, it is buffer, and that is not a question mark at the end. Buffer, definitely. A buffer, yes. Organic molecules must contain this element. What does the word organic mean? Just give me the letter. What must they contain? Yes, carbon. Organic molecules means lots of carbon. That's what the word organic means. All right. Monomer that makes up proteins. Notice I tell you we're going to do it about 500 times. Yes, amino acids. You'll probably see it every week from now to the end of the class. So nobody should be telling me that something else makes up proteins. Amino acids. All right. Now, last one deals with your suffixes. Lactose, OSE. What type of molecule is it because it ends in OSE? Yes, it is a carb. And lactase, if it's ASE, what is it? I said that briefly. Maybe you guys picked up on it when you did your assignments. ASE means it's a what? First, you can give me type of molecule, and then you can tell me more specifically if you know what you're doing. Lactose is the thing that lactase acts on. That's why they sound similar. It is, no, lactase is not a sugar. It is a protein and it's an enzyme. That's correct. All enzymes end in ASE. That is your clue. And if it's an enzyme, you know the type of molecule is protein. That's where that's coming from. So look for that on your test. Know that ASEs are enzymes, and please know what enzymes do. All right, I'm going to go through the Quizlet now, and then I'll post it. You can look at your study guide to follow along with me, but I have changed all of the study guide questions into a Quizlet. All right, we start with, hopefully they match up exactly. I'm doing number 1, how are monomers and polymers different, and provide an example of each. I'll give you my answer. You don't have to answer all of these exactly the way I do. Just understand what I'm talking about. Monomer is the building block of all of these things. Think of it as a brick. A monomer is a single brick. Polymer is when you put those bricks together. And what I wrote was a monomer is a single molecule. Polymer is a string of monomers bonded together. Glucose is a monomer. That's the easiest one to remember. And cellulose is a polymer of glucose molecules. We did talk about cellulose last time. I will stress it again. You must know what cellulose is and why it is special. We cannot digest cellulose. It comes from plants. Cellulose makes up the cell wall of the plant, which is the next unit, which we're doing. And since it ends in OSE, it is unique because it's a carb, a sugar, that is forming the structure that holds a plant cell together. That is the only instance of that. Remember, our cell membranes are made of lipid. All cell membranes are made of lipid. The exception is the plant cell wall made of cellulose, which we don't digest. Now, starch, we discussed what starch means. Starches are complex glucose molecules put together. It's polymers of carbs. And when we store our glucose, short-term carb storage is starch. For an animal cell, it's called glycogen. For a plant cell, it's called cellulose or starch. And we don't break it down. So you will get a cellulose question. Even though it's a bank of questions, I can guarantee you're going to see some cellulose. They'll ask you what kind of molecule it is. They might ask you what it does and why it's special. All right, what type of organic compound is cellulose? I just gave you the answer. Organic compound is which macromolecule? It's a carb. And it's used in plants to provide structure for the cell wall. All right, what type of organic compound is glucose? We just did that. Carb. You know it's a carb, and we use it for short-term energy. It's the monomer of carbohydrates is glucose. And why is glucose so important to organisms? Generally, just asking you, what do we use it for? Primary energy source. Primary energy source. List the polysaccharides commonly found in animals or animal cells. And I just gave you that. Glycogen. Now, if you guys need to go through these cards and test yourself, feel free to use it as flashcards. Feel free to add to your notes if you need to before the test. But the polysaccharide, which is our version of starch for animal cells, is glycogen. Now, the polysaccharides commonly found in plants or plant cells. Okay, already told you that. Starch. Storage polysaccharide commonly found in plants is starch. Do you have to turn in the study guide? No. When we were in school, that would be an actual assignment that I would grade. I mean, you'd be an idiot not to do your study guide, so I'm not going to make it an assignment. I don't have to wonder whether you guys are doing it or not. Trying to take this test without doing the study guide would be kind of foolish. So, no. However you do it and whatever format it takes for you, just hold on to it. Which organic compounds are sources of energy to organisms? All right, it sounds like a tricky question, but what things do we use? The four macromolecules, which ones are used for energy? It's carbs and lipids. Remember, short and long term. What are the components of most lipid molecules? We just did that in the warm-up. Fatty acids and glycerol. What are the negative health effects related to excess lipids in the body? When you did the interactive slides, showed you the picture of the butter. What's bad about having excess lipids in the body? You get heart disease. You can get clogged blood vessels from those excess lipids. Remember, at certain temperatures, those saturated fats want to turn solid. Now, they might, even if they don't get to be as solid as the butter in your kitchen, it still blocks up the works. You have excess of that you're not processing, you can have heart disease. What is the function of nucleic acids and how is this function accomplished? Nucleic acid, Na, you know your NAs are your R and DNA. And their main thing is holding all of your genetic information. Those sequence of bases, A, T, G, and C, it's only 4 of them, but the sequence that they're in throughout your DNA is what gives you your different instructions. So the sequence of the bases determines their instructions. You take those 4 and you mix them up in all different kinds of ways, and it tells the body what to do. It is your blueprint for everything. We will be going over that many times. DNA is the master blueprint for everything that your body is going to do. It gets its instructions from the DNA. And what differentiates one protein from another? All right, this one we have not gone through in a lot of detail. I did not teach it to you directly, but it was in your stuff. The proteins are made up of amino acids. And just like the DNA, the sequence of those amino acids is what makes one protein different from another. It's like the letters of the alphabet. There's only 20 amino acids, but we have a ton of proteins. What we end up doing is rearranging those 20 things, like the way we rearrange letters in the alphabet, and you can make thousands and thousands of words. You take those amino acids, put them in different sequences, and you're making different proteins. What type of organic compound is hemoglobin? We did that on our last class. Hemoglobin is a protein, carries oxygen for the blood. And if your body is in need of amino acids, what type of food should you eat? Okay, Easy. If you know what that's just another way of asking, do you know what amino acids make? If you know that the macromolecule is protein, all of you know the different things that you can eat to get protein. Meat, beans, eggs, numerous answers. So they will ask you something like that in some way. That's the concept. The question will be based on it. Create an analogy for the enzyme substrate relationship. That one, somebody type it in the chat box, because it's the thing they tell you all the time when they say they're teaching you about enzymes. What analogy do they give you? They work like what? Yes, M.A.P., lock and key. That is the main analogy you get for enzymes. They work like a lock and key because the enzyme, the shape of it, will only work for one substrate based on how they fit together. Just like every lock has a key with a certain shape that can open it. And how does specificity relate to enzymes? All right, very weird worded question, but what they want to note is that each enzyme has a shape that only allows it to fit one specific substrate. That is your specificity. One enzyme, one substrate. Now, what can cause them to denature? Also in your warm-up. You see how valuable these warm-ups are. If we go through this stuff, it's like I don't have to even give you a lecture. The stuff comes up. We talked about it. Gabe gave us the answer before. Temperature extreme and pH changes can denature or change the shape of a protein. That's what that word means. What are the effects of denaturation? We just answered that. Enzyme will lose its shape and can't function any longer. We broke this down because remember we said we looked at the test questions and just converted them to concept questions. So if you just saw that I asked you those two things and they're related to each other, that means that's more than one question on the test. What happens to the rate of an enzyme reaction if temperature increases? In general, when you have chemistry and you heat something up, what happens to the rate? Increase or decrease? Anybody?
Increases.
Right. In general, heating something up will increase its rate. Now, everything has its limit. You'll see that if we were looking at a graph, heating up the enzyme reaction will make that curve start to go up, but it has a peak. When it hits the peak, then it starts to go down. Now we're getting into denature territory because it's too hot. So increases in general, but when you reach the peak, you're starting to damage it and you're diminishing returns. All right, draw a nucleic acid and describe its components. I gave you that interactive slide with that. You should be able to do a very, very basic drawing of a nucleic acid. A circle for the phosphate, one of those hexagons or ring for the sugar, and then over to the side, we usually just make it a generic rectangle and put a letter in it. That's the base, A, T, G, or C. Phosphate, sugar, base. If you saw one, you should be able to recognize it and tell the different parts. Remember, the phosphate and sugar are the things you hold on to with your hands on a ladder. The bases, the A, T, G, and C, is where you would put your feet to climb up. All right, use Chargaff's rule to determine the amount of A, T, and C in a sample if it is 30% G. All right, if it's 30% G, somebody tell me how much C I have. I know that immediately. How much C do I have if I have 30% G? Yes, 30.
70%. Oh.
Yeah, see, which is why you should always put yourself on delay. Your brain needs the time to function. Yes, 30% G means 30% C because they go together. The companion base is of equal amounts, which means the other bases, what I have left is I had 30 and 30. I have 40 left for A and T, so I need to split them. So how much A do I have now? Yes, Leah, 20. 20, because remember, any of the ones that don't go together have to equal 50. All of them have to equal 100. Chargaff's rule All you have to do is know one of them and you can figure out the other three. So somewhere on the test, you're going to get one question where they just want to know, do you understand how that works? All right, given the DNA sequence, TAG, GGC, ACT, and TGG. Now, notice that they're in triplets. Those are called codons. We will get to that later. I'm not going to give you more than you need. But they always show DNA and RNA sequences in sets of 3. And all you're doing is using the pair rule. If it's the complementary strand, they're asking you for each triplet, you are going to write a triplet underneath it that goes with it. So let's just do the first 2. If I have TAG, what is the strand that goes with TAG? What would that look like? Yes, it would be A to go with the T to go with the A, and C to go with the G. So it's ATC would be the complement. That's first codon. So you just do that all the way across and look at your multiple choice answers and find the right one. Please make sure you go all the way to the end. If they get tricky on you and they do all like 3 or most of the codons are together, somebody might skip and say, okay, I know which one it is. That's the one. Make sure you've looked at all the answers and you go all the way to the 4th codon if they give you a strand to do the complement. That's why it's called the complementary strand, because they fit together. Very easy stuff. Last one. Compare and contrast the structures of DNA and RNA. That was in the Edpuzzle that you guys had. The Amoeba Sisters went through that. Hopefully you guys did those. I will recommend one more time. Anytime I give you an Amoeba Sisters video, sometimes it will be a choice with something else. Do yourself a favor, and even if you didn't pick it to do the assignment, go back and watch the Amoeba Sisters video. I'm going to be forced to give you fewer of them because they have Amoeba Sisters for every single little subject matter in biology. I'm going to have to pare that down because you guys don't have time to do all of them. So I'm picking the most important ones and sticking them somewhere, either in a hyperdoc or giving it to you by itself. Do your Amoeba Sisters. They're only a few minutes each, but they are important. The one on DNA and RNA told you that DNA is a double-strand helix. Your classic picture of a DNA molecule is taking a ladder and twisting it. That's 2 strands put together. That's why it looks like that. The 2 strands are the sides of the ladder, and they twist. All right, and they have the bases A, T, G, and C. RNA is a single strand. Picture taking a ladder and chopping it right down the middle. So now I have half a ladder. That's my RNA, but it has the bases A, U, G, and C. There's no T in RNA. Knowing that concept is the basis of 1 or 2 questions on the test. They might ask you about the makeup of DNA versus RNA. They might ask you the bases, the difference, knowing that the base U, uracil, is only found in RNA. I know that's a question in there in the bank somewhere, and I've told you that a few times. If I see uracil, I'm dealing with RNA. So know those differences in their structure, what they look like, and the bases that make them up. We will get into the differences in what they do later on when we get into making protein. But for right now, that's what you need to know for the test. So I think that covers everything that you guys will see. The last thing I heard was that they were definitely thinking about adding 1 or 2 short answer questions at the end, just to make sure that people aren't strictly cheating. We know we can't stop cheating. If somebody wants to do it bad enough, they can cheat on the test. But we're just making it so it's not worth your while. Between the test bank, the timer, the short answer part, just know your stuff and go through, and it should be easy. There's no need to cheat. But if you did, eventually that was going to come out. With the kinds of questions we're asking you, sooner or later, if you don't really know this stuff, it's going to come back on you. So do your best. Try to know the concepts because we're going to build on them. This course, until we get to the end of biology, everything we're doing will get reused. So if you don't know Unit 1, it will come back to bite you again later on because I'm going to be building on the stuff we just talked about. All right, ask your questions now. Otherwise, I'm going to let you guys go finish your assignments and take your quiz.
Where do I access the notebook? Student.
I was not specific enough. Anybody but Andrew that has a question. The notebook is in its own section, Andrew. Look near the top. You'll see a section that says notebook.
I
And-
only did some of the vocab in it.
Okay. Anybody besides Andrew have a question? Yes, Jada, I got you. Anybody else have a burning question?
So, will you be posting the Quizlet after this?
Yeah, as soon as you guys, in fact, as soon as we get off this call, I'm going to do it right this second.
All right, thank you.
Anybody else? Look for the boom cards, the macromolecules boom cards. It will be, different ways of asking this stuff other than the concept review. If you just want to take some time after you've taken the post quiz, because remember, you can take that more than once. Between that, the study guide, if you say you just want one more thing to go through to help you study, you can use the boom cards, but it's not an assignment, not for a grade. Just giving you those, because if we were in class and we had full-time classes, I would have given you that as something to do for a grade. There was an escape room that I was going to give you guys, but I'm not going to. I'll hold it till the end of the year, the semester, I mean, and we'll use it later on, because I think you guys have enough that you need to finish and study to get ready for the test. So not going to do that. All right, if you guys don't need anything, I'm going to stay on the call. You can drop off if you don't need me. Anybody that does need me can either stay on or get back in later and ask me what you need to ask, but I'm going to post your Quizlet now. And I will also post